Good day, Ma'am Alexi. So today we will going to report about the immunotechniques, its interaction, and process. Right now, I am going to report about or discuss about the immunoprecipitation. So immunoprecipitation is an antigen or antibody interaction or binding that highly specific that occurs in a similar bimolecular enzymes and substrate. So the binding between the antigen or the AG and antibody, which is the AB, is weakly interaction that has a weak interaction and reversible that occurs or consists in a different forces, that is van der Waals forces, hydrophobic forces, electrostatic forces, and H bonding. So there is what we call the small area of bonding, which is called the epitope or the antigen determinant. So this bonding do have interaction when you combine the antigen or antibody bonding. The smallest area of an antigen capable of combining with the antibodies is called the antigen determinant or epitope, while the smallest area of the antigen molecules and combining with the epitope is called the paratope. And then the surface of the many epitope is called the valence. So valence determines the number of antibody molecule that can be combined with the antigen to have an interaction together. However, if there are one present of an epitope that can be found in the antigen, it is called a monovalent. But if there are various number of epitope that can be found in the antigen, that is termed as polyvalent. This is the interaction of immunoprecipitation that have a different insoluble reaction. Therefore, immunoprecipitation is a result of interaction combining with a soluble antigen and soluble antibody for it to be formed in insoluble complex in which antibodies aggregates with soluble antigen, and it is called precipitants. And this reaction can be performed in a solution or in gel to have an outcome reaction.
So hi guys, welcome to my video, and today my topic is all about the immunoprecipitation reaction in gel. So what is immunoprecipitation reaction? So immunoprecipitation reaction, or IP, is a widely used technique in molecular biology and biochemistry to isolate a specific protein or complex of protein from mixtures using antibodies. So IP is a technique that used in various reasons, just like in the description below, it says that it isolate proteins, or complex protein for mixing using the antibodies. So the procedure involves formation of antibodies complex, followed by the precipitation of the complex separation of the target protein from the rest of cellular components. So gel electrophysics is often used the subsequent step to analyze the visualize the immunoprecipitated protein. So here's the general overview of the immunoprecipitation reaction and its analysis in gel. So immunoprecipitation reaction. So antibodies incubation. So antibody specific is to target proteins and incubate with a cell lysate or protein samples containing the protein interest. So next is the protein AG or beads. Protein A or protein G beads are commonly used to capture antibody antigen complexes. So protein A and G binds to the Fc region of antibodies and facilitating the precipitation of the entire complex. So next is the washing. So unbound proteins are removed through washing. Steps reduction and specific interaction. Next one is the elution. The immunoprecipitated complex are eluted from the beads. Often use a low pH buffer or a competitive elution buffer. So next one is the gel electrophysics. Sample Sample preparation. The eluted samples are denatured and prepared for gel electrophysis by adding the loading buffer containing SDS or sodium dosyl sulfate and merfatotanol. So SDS denatured proteins and imparts negative charge and proportional to the protein masses. So next one is the loading onto gel. So the samples is loaded into uh, wells of the STS phage or the polyclymate gel electrophysis gel. So the gel is run under the electric field, causing the protein to migrate based on size. Next one is the electrophysis. So the protein migrates through the gel different rates with smaller proteins moving faster than larger ones. So the fourth one is the protein separation. So the protein separation, the gel is run until the proteins are sufficiently separated according to their molecular weights. Next one is staining, no? The gel is stained the gel is stained by dye, no? Example, Comasis blue to visualize the proteins. So next one is the Western blotting. So this is optional. So following electrophysis, protein can transfer to a membrane for Western blotting. This allows the detection of the specific protein of interest using secondary antibodies coupled to the detection system. So last one is the analysis. So analysis, bands of the gel membranes are analyzed to confirm the presence of the target proteins in the precipitated sample. By combining immunoprecipitation with gel electrophysis, researchers can specify isolated and analyzed protein of interest from complex mixtures. So the technique is valuable for studying protein-protein interaction, post-translational modification, and assessing protein levels in various experimental conditions. So that's all. That's all about the immunoprecipitated reaction in gel. So that's all, and thank you.
Good afternoon, ma'am. Good afternoon, classmates. I am Inchimi Heraldi. I am going to report about radial immunodiffusion or Mancini method. So, what is radial immunodiffusion? It is also known as the Mancini method, is a laboratory technique used to quantify the concentration of specific proteins in a sample. This method is based on the principle of antigen or antibody interaction. Okay, so the relative concentration of an antigen can be determined by an by a sample quantitative assay in which an antigen sample is placed in a well and allowed to diffuse into agar gel containing antibody. So, in agar gel, antibody is uniformly distributed at the region of equivalence. A precipitation ring forms around the well. The diameter of the ring is proportional to the log of the concentration of antigen since the amount of antibody is constant. So here's a simplified explanation of how it works. First, a specific antibody is mixed with a gel, usually agarose, and allowed to diffuse evenly throughout the gel. This gel is then poured into a petri dish or similar container to form a thin layer. Second, once the gel has set, small wells are cut into it, and a sample containing the antigen or protein of interest is added to these wells. Okay, so third, the antigen diffuses out of the well into the gel where it binds with the antibody. This forms a visible ring or zone of precipitation in the gel. And the last, The size of this ring is directly proportional to the concentration of antigen in the sample. By comparing the size of the ring to a standard curve created using samples of known concentration, the concentration of antigen is the unknown sample can be determined. Next is partial identity. It occurs when two antigens share some epitopes, but one of the other has a unique epitope. Okay, let's see what is in figure 10.3. Okay, so this is an Austerlony double immune diffusion patterns. Okay, antibody that is a mixture of antibody 1 and 2 is placed in the central well. Okay, and unknown antigens are placed in the outside wells. Okay, in letter A, which is identity, the arc symbol indicates that the two antigens AG1 and AG2 are identical, and letter B, which is the non-identity, two cross lines represents two different precipitation reactions. So the antigens share no common epitopes. And letter C, which is the partial identity, AG1 and AG2 share some epitopes. Okay, let's proceed to the Auster-Loney method. It can also be used to estimate the relative concentration of antigens, which an antigen has a relatively higher concentration. The equivalence zone will be formed a little bit away the antigen well. So, when an antigen has a relatively lower concentration, the equivalent zone will be formed a little bit closer to the antigen well. So, next is the agglutination reactions. So, agglutination is the visible clumping of a particulate antigen when mixed with antibodies specific for the particulate antigens, and agglutinins are antibodies that produce such reactions. So the general term agglutinin is used to describe antibodies that agglutinate particulate antigens. When the antigen is an erythrocyte, the term hemagglutination is used. All antibodies can theoretically agglutinate particulate antigens, but IgM, due to its high valence, is a particularly good agglutinin. Agglutination reaction is similar in principle to precipitation reactions, just an excess of antibody inhibits precipitation reactions, such as excess can also inhibit agglutination reactions. This inhibition, called the Poisson effect. So, the Poisson effect, excess of antibodies in a serum sample, can exhibit the antigen-antibody interaction and subsequent agglutination reaction, leading to a false negative result. And in this case of antigen excess, the post-zone effect occurs. Post-zone effect, a false negative test caused by an excessive antigen relative to antibodies, which prevents antibody to antigen cross-linking. So there are two forms of agglutination. First is the direct and active agglutination. The antigen occurs naturally on particle. And second, passive indirect agglutination. The antigen must first be bound to an inert particle to detect an antibody. Okay, for direct or active agglutination, when the antigen is an integral part of the surface of a cell or other insoluble particle, the agglutination reaction is called direct agglutination. For passive and direct agglutination, The agglutination rate only works with particulate antigens. However, it is possible to coat cell or other insoluble particles with a soluble antigen, example viral antigen, a polysaccharide, or a heptane, and use the coated cells in an agglutination test for antibody to the soluble antigen.
So hello everyone, especially to you Ma'am Alexi Packet. So for my assigned topic, I am going to discuss about the direct ELISA and the indirect ELISA. So first, I am going to discuss about the direct ELISA. So the direct ELISA is one of the variant of enzyme link immunosorbent assay, or in short, ELISA. It is an antigen in which it is directly captured by specific binding of antibody, that the antibody is conjugated to an enzyme which can turn a colorless substrate into a colored product. So direct ELISA is one of the type of an assay. So in direct ELISA, the antigen is immobilized on the bottom of the microtiter plate. On the right of the sample contains the target antigen, which is the orange. So the left serves as the control and contains only the proteins. So now the antibody solution is provided. The specific antibody is designed to recognize antigen and forms non-covalent band. After washing it, it remains bound, whereas the antibody is washed out in the control or in the left side. So now the substrate is given by the antibody to bound. The antigen is conjugated to an enzyme. This enzyme is converting the substrate to detectable product, which lead to a color change. So the intensity can be measured using the spectrophotometry. So now let's proceed to the indirect ELISA. So in indirect ELISA, it is the second types of ELISA. In indirect ELISA, the antigen is mobilized similarly, but instead of directly adding the enzyme link antibody against the antigen, a primary antibody that binds to the antigen is used. So then a secondary enzyme link antibody that binds to this primary antibody is applied. This secondary antibody enables the detection of the primary antibody and consequently the presence of the antigen. So the components of the samples are given time to adhere the plastic of the wheels looking right the sample. So contains the target molecules, which is the orange. So it is all. So in shown in the left, it contains only the specific protein, same as the indirect ELISA. A solution is containing antibodies, and antibodies is provided primary antibody is specifically designed to recognize and bind to the antigen. After washing, antibody remains bound. Washing removes the antibody, and in the control, secondary antibody is given to each well that one binds the remaining primary antibody. The washing steps removes the unbound antibody in the control. Now the wells are filled up solutions containing the substrate. The enzyme link the secondary antibody converts the substrate to a product which leads to a change of color. So again, the intensity can be detected through the spectrometry. And that would be all. Thank you.
I am Giselle Jalapon, and my assigned topic is all about the competitive ELISA and sandwich ELISA. So what is sandwich ELISA? Sandwich ELISA, or enzyme-linked immunosorbent assay, is a type of immunoassay used to detect and quantify the presence of specific proteins or antigens in a sample. It is based on the principles of antigen or antibody interactions. In a sandwich ELISA, the target protein or antigen is captured between two layers of antibodies. The process involves several steps. First, coating. The first step is to coat the surface of a microplate with a capture antibody that specifically binds to the target protein or antigen. Second, blocking. After coating, the microplate is blocked using a blocking agent such as bovine serum albumin or BSA or casein to prevent non-specific binding of other proteins. Third, sample additions. The sample containing the target protein or antigen is added to the microplate and allowed to incubate. If the target protein or antigen is present in the sample, it will bind to the captured antibody. Fourth, washing. After incubation, the microplate is washed to remove any unbound substance. Fifth, detection antibody. A detection antibody is added to the microplate which recognizes a different epitope on the target protein or antigen. This detection antibody is usually labeled with an enzyme such as horseradish prosidase or HRP, which allows to detect the detection of the bound antigen. 6. Substrate addition. A substrate specific to the enzyme's label is added to the microplate. The enzyme on the detection antibody catalyzes reactions with the substrate, resulting in a measurable signal, example color change. Last, the signal measurement. The signal generated by the enzyme substrate reaction is measured using a spectrophotometer or other appropriate detection method. The intensity of the signal is directly proportional to the amount of the target protein or antigen present in the sample. So sandwich ELISA is commonly used in research and diagnostic laboratories for the detections and quantifications of various proteins, including cytokines, growth factors, hormones, and viral antigens. It offers high specificity and sensitivity, allowing or accurate measurements of proteins concentration in biological samples. ELISA. Competitive ELISA is enzyme-linked immunosorbent assay is another type of immunoassay used to detect and quantify the presence of specific proteins or antigens in a sample. It is based on the principles of competitions between a labeled antigen and a labeled antigen for a limited number of antibody binding sites. So, in a competitive ELISA, the target protein or antigen in a sample competes with the labeled antigen or a labeled antigen analog for binding to a limited amount of specific antibodies that immobilize on the surface of a microplate. So, the process involves several steps. First, coating. The first step is to coat the surface of microplate with captured antibodies that specifically bind to the target proteins or antigen. Second, blocking. After coating, the microplate is blocked using a blocking agent to prevent a non-specific binding of other proteins. Sample addition. The sample containing the target protein or antigen added to the microplate and allowed to incubate the target protein or antigen is present in the sample. It would compete with the labeled antigen for binding to the limited antibody sites. The greater the concentration of the target protein or antigen in a sample, the less labeled antigen will be able to bind to the antibodies. Fourth, washing. After incubation, the microplate is washed to remove any unbound substances. Detection. A detection reagent is added to the microplate which contain an enzymes that can generate measurable signals. The detection reagent typically consists of antibodies that recognize and bind to the labeled antigen. 6. 6. Substrate additions. A substrate specific the enzymes label is added to the microplate. The enzymes on the detections reagent catalyze a reactions with the substrate, resulting in a measurable signal, example the color change. 7. Signal measurement. The signal generated by the enzyme substrate reaction is measured using a spectrophotometer or other appropriate detection method. The intensity of the signal is inversely proportional to the amount of the target protein or antigen in the sample. High concentration of the target protein or antigen in a sample will result in a weaker signal as they will bind more antibodies and leave fewer binding sites for the labeled antigen. And competitive ELISA is commonly used in research and diagnostics laboratories for the detection and quantifications of various proteins, molecules, hormones, drugs, and environmental contaminants and offer high sensitivity and specificity and can be used to measure a wide range of target analysis. And that's all. Thank you.